All right, we're on. So, welcome everybody to the fifth edition of the Multiomics Reading Group. I'm your host for today, Cesar Miguel Valdez Cordova, and today it is my absolute pleasure to introduce a longtime collaborator and friend, Aaron Wentler. He is a PhD student in AI for drug discovery at the Queen Mary University of London in the group of Michael Barnes. His research mainly focuses on combining multiomics data and machine learning methods to improve early-stage drug discovery. Today, he will be presenting PertEval-scFM, benchmarking single-cell foundation models for perturbation effect prediction. Without any further ado, Aaron, take it away.
Yes, thank you, Cesar, for the wonderful introduction. Longtime collaborator Cesar actually is a big part in this project as well that I will be presenting today, PertEval-scFM. This is a benchmarking study in which we looked at single-cell foundation models in a zero-shot context, and particularly how they would fare for the task of perturbation effect prediction, which is generally one of the more challenging single-cell prediction tasks. But before we get to that, for the people that might not be intimately aware with perturbation prediction, let me just try to put it a bit into perspective what we're actually looking at here and why we care about it. So, as you probably are aware, if you are working in biology with multiomics data, there have been some tremendous improvements in experimental methods when it comes to transcriptomics, particularly single-cell RNA sequencing. Nowadays allows us to look at transcriptomes on a single cell, really on a cell-to-cell level, which is very interesting, and combine this with sort of another experimental breakthrough of CRISPR-Cas9 or other sort of similar CRISPR modalities that allow you to actually really accurately target individual genes and perturb them either which way, activating them or inhibiting them. Combining these two technologies gives you sort of a very powerful tool to look at transcriptomic and gene regulatory mechanisms. And there's going to be lots of research going on trying to understand the biology from this lens. And perturbation prediction is sort of one small part of this, where we're trying to actually understand this sort of landscape perturbation space and be able to predict on it before you actually even do such experiments. And we'll see a bit later on why that might be interesting. So, sort of an important concept I need to introduce, because that's mainly what we're looking at in our study, is that of perturbation effect. So for those not aware of Perturb-seq experiments, generally how these experiments get carried out is you have a set of control cells, and you measure their transcriptomes, then to a similar set of cells, but importantly not the exact same cells, but just the same cell types. You apply a perturbation, and again you measure the transcriptome, and now you can compare between the perturbed state and the control state, take the difference, and there you have your perturbation effect. So in this example here, we might perturb gene X, which has sort of downstream effects, in this case on gene A, which is slightly inhibitory, and gene B, which is slightly upregulating. And this gives you sort of a perturbation effect distribution. Now, why would we care about this? Like I said before, looking at the transcriptomes, it gives you sort of an idea of biological mechanisms at the RNA level. Moreover, it gives you an idea of how genes interact with each other and how they form parts of these sort of bigger gene regulatory networks, which oftentimes actually underpin lots of diseases. And being able to understand these mechanisms for one and intervene in them, maybe more importantly, would allow us to more effectively find potential treatments for certain diseases. How that might facilitate is, for example, think about perturbation prediction. You have some cell state which is diseased. Now you want to know, how do we push this cell state back into a sort of healthy state? Is where you could use, or you could envision some sort of perturbation simulator that can tell you exactly, okay, if you target this gene, And oftentimes, as you might be aware, genes or their corresponding proteins are the drug targets for the eventual proteins or compounds that we use to cure diseases. How can we target them to shift this disease state back into a healthy state, as in this case parameterized by the transcriptome? And in turn, looking at it like this from a sort of very detailed mechanistic level will then also allow you to distinguish between even individuals, but more realistically, for example, between populations, and would allow us to move a bit more towards a paradigm of personalized medicine. Okay. Now that we've sort of put into context perturbation prediction and give a quick rundown on that part, let's move towards the machine learning side, where very recently we've seen this big push for what is come to be called foundation models, which you can characterize as sort of very big, self-supervised, pre-trained models that can be fine-tuned for all sorts of various tasks. And these models have shown tremendous efficacy in various tasks, most top of mind being large language models. Excuse me. I've got a slight cold I'm dealing with. As I was saying, most top of mind being large language models. Begs the question, could we sort of use these types of models for maybe not equally large, but other data types, like, for example, single-cell RNA sequencing, in this particular case of single-cell foundation models. So the way you'd go about it in this particular case is you'd have your big, what we call in the field of cell-by-gene matrix, where you have in your rows all sorts of different cells from sort of different states or different types. And in your columns, you have the genes with their corresponding expression values. Then you pre-train on this data set by some sort of masked learning objective, by which you try to reconstruct the expression values by masking them out, sort of looking at the local, and with sort of transformer architecture, it also allows you to look at a global context of expression values, and from this try to reconstruct the expression value that was missing. And it is argued that this process of pre-training then allows you to calculate cellular embeddings of cell states that nicely distinguish very sort of in a resolved way onto cell states or cell types. And these embeddings can then consequently be used for all sorts of different tasks, like cell type annotation, data integration. And importantly for us, it has been said that these can also be used for perturbation prediction. Now, I think it is always important when using these types of models, you consider the assumptions sort of underlying them. And there are always sort of three in this particular case that are very top of mind for me. There are, of course, more. But when you think about why a certain modeling paradigm might or might not work for a certain use case, I think it's always good to go back to sort of the basics and the underlying assumptions here. In this case, sort of this biological application of trying to model transcriptomes. So first and foremost, the way we model biology in this case with these big Perturb-seq single-cell RNA data sets is kind of as cells are bags of RNA. And if you know even the slightest bit about biology, you know that this is a very overly simplified view of what's actually going on in a cell. So even though you might be able to understand mechanistically on a transcriptomic level what might be going on, this might not always tell the full story. So it's a very important thing to keep in the back of your head when proceeding with these types of studies. Secondly, these foundation models often rely on some sort of transformer backbone. And these models, in their sort of best use cases, oftentimes are being applied in some sort of context in which they operate on sequential data. Whereas these big cell-by-gene matrices that I used earlier are not necessarily of this nature. I mean, think about language, right? You have these sentences in which they consist of words, and these words have some very strict sequential semanticity associated with them that makes a sentence what it is. Oftentimes in single-cell RNA-seq, you might see people make this analogy with the cell being a sentence and then the genes being the words. But this analogy kind of falls flat a bit if you consider that in these types of RNA-seq data, the order is not necessarily as profound as it would be in language, or even in keeping it in biology space, something like AlphaFold, where amino acid sequences, the ordinality there of the individual amino acids is crucially important to determine the final 3D structure conformation of the protein. Finally, it is argued that by using a self-supervised MLM objective, mass language modeling, the model learns what certain gene interactions entail. So the upregulation of one gene, how would it affect another gene? And by just exposing your model to many of these examples, it's argued that the gene interactions gets very, that they get represented very well in embedding space. And these are just sort of the underlying assumptions that I always think put into perspective very well why you might want to apply a model like this in this context, and then also help us explain what we see on sort of the result side come that time. But let us not get ahead of ourselves for a little bit. First, let's sort of introduce why we might want to benchmark single-cell foundation models for perturbation prediction, and what sort of our own intuition was and the reason for why we went down this path. Because if you have been following this particular branch of foundation models combined with single cell data, you might have seen a slew of these papers come out recently, where sort of the angle is slightly different, but the overall narrative arc is looking at how useful are these single cell foundation models for various tasks amongst Swiss perturbation prediction. In our case, we were particularly interested in the zero-shot embeddings of the single-cell foundation models, kind of sort of analogous, if you are familiar with large language models. After their pre-training stage, oftentimes to check the quality of the embeddings, so-called linear probes or even nonlinear probes are applied straight away to zero-shot embeddings on some sort of interesting task. In the language case, that might be some sort of classification. To see how sort of inherently informative or information-rich embeddings are. And analogously, we kind of wanted to probe the cell embeddings in that way for perturbation prediction, where it's not necessarily classification, rather a regression onto a gene expression, post-perturbation gene expression vector. And secondly, since we do a benchmarking study, this of course begs the question: how do we fairly and properly evaluate transcriptomic perturbation prediction, which is a lot more of a contentious topic than it might seem at face value. You can very easily misrepresent perturbation prediction performance, even inadvertently. So it's important, crucially important, that you think very well about how you present and with metrics you choose. Finally, of course, then, given all of this together, how might we improve the next generation of foundation models for perturbation prediction? Or even is it useful to continue down this path of research, given what we find? Okay, let's get into our method. There are several steps in PertEval that compose PertEval. The first of which is our pre-processing module. This is fairly standard. If you work with single cell data, you have probably seen and heard and done this many times. And the pre-processing steps. What I think is important to highlight here is our choice of baselines. We have sort of a calibration baseline in the mean expression. So what you do is, for a given cell, you calculate the mean expression across all genes. And this will tell you kind of a control, sort of to calibrate literally your prediction by sort of predicting the input back. So what would your metrics say if we predict very little change on the input side? And oftentimes, interestingly, depending on sort of your setting, this is already a very good initial guess, if you will. And why that is, I'll explain a bit later with a figure. Secondly, to similarly sort of anchor or calibrate our embedding models, we chose to also have a baseline in which we do not embed our gene expression values, but use them straight away to sort of, if there is sort of a performance delta between the embeddings and the raw gene expression, you could kind of quantify what then exactly is the sort of extra semanticity that's been introduced in the embedding space through these foundation models, if there is any at all to begin with for perturbation prediction. Right. Another crucial step is to decide how you represent a perturbation in silico. And we kind of have two modes. For a raw expression baseline, you might argue you could do something like a one-hot encoding, which we tried, but very early on we figured out that actually a very interesting setting for perturbation prediction is to look at unseen perturbations. So you train on a set of perturbations in your training data. And when I say perturbation, because we're talking about CRISPR-based genetic perturbation, we're really talking about one gene in this case. So in sort of the one-hot encoding case, you would sort of have this lookup for each gene, one-hot encoding, and then you would expose your model to whichever perturbation are in the training set, and that would help it then identify what perturbation it is right now working with. However, this does not really allow you to extend to unseen perturbations, as you would not have the one-hot encoding at that stage, in this sort of lookup training stage. By doing the Pearson correlation, you already introduce some sort of first-order interactions between different genes. And so the way in which you do it is by having your expression vector for a given perturbation, or actually however many expression vectors you have for that perturbation, because of course you measure multiple cells. And then you calculate the correlation for the perturbable gene with itself, so the autocorrelation, so that always gets assigned 1. And then you also calculate the expression for the perturbable gene with all the other genes, sort of indicating a first-order interaction strength for that gene with other genes. So that also already then gives the model some sense of how genes relate, even if it's never seen a perturbation in that other gene. And to us, that seemed a bit more fair than just some sort of one-hot encoding based method. For our single-cell foundation models, we did slightly differently. We did nullify the gene expression, which literally means setting the expression to 0, sort of knocking it out, if you will, and then generate an embedding from this knocked out version or nullified version of your data, where it is crucial to note that between single-cell foundation models, this process is slightly different. For example, some foundation models represent their gene expression non-parametrically. For example, GeneFormer takes the gene expression data, it uses the numerical values to rank each of the genes, and then assigns some sort of gene token, and then you have sort of a ranked vector of gene tokens, where the one at the top of the vector indicate very highly expressed genes, the one at the bottom very lowly expressed genes. What nullification then entails is pushing down your gene from wherever it was to the end of the vector. Whereas parametric foundation models like SCGPT that directly use the numerical expression count, you can just literally set that value to 0. Then for training, we take a slightly non-conventional approach for generating our trained test splits. We got tremendous help from Jascha Ektavaya over at Marinka Zitnik lab, who developed Spectra. And Spectra is a graph-based method to sort of generate trained test splits where you control for what in the method of Spectra is called cross-split overlap. So to explain that in the context of perturbation prediction, what you do is you calculate similarities between perturbations, then you map these similarities to a graph where sort of perturbations that are spatially close to each other in this graph are similar. And then you start to drop out edges between these nodes. And this then leads to subgraphs. Then you can sample from these subgraphs into a train set. So from the same subgraph, you would sample into the train set, and then from the same subgraph, you would sample into the test set. And this gives you some sort of guarantee on the similarity between your training and your testing set. And the parameter that decides how sparse your graph is, we call the sparsification probability. So the higher this parameter is, the more dissimilar your train and test sets are, and thus the more difficult your eval is. And oftentimes when you somehow try to do a train test split, this is kind of what you're trying to get at. Oftentimes you parameterize it in different ways. You do it through time or some sort of other maybe biological meaningful way. But in this way, you sort of directly quantify actually the data you are working with, and I think it's a really elegant way to defining train test splits. So I would encourage anyone to check out Yasha's work on spectra, which recently got published in Nature Machine Intelligence. Now, given these train test splits, we again take an inspiration for a large language model field in which you probe on zero-shot embeddings. We use a simple one-layer MLP probe to extract or to regress onto our post-perturbation expression, perturbation effect. Yeah. Now, benchmarking study. So there has to, of course, be some consideration into evaluation metrics. And for perturbation prediction, your selection of evaluation metrics is incredibly important into how you're going to actually interpret what's going on. And we developed together with Yasha, or sort of tweaked, I should say, the area under the spectral performance curve specifically for this task here of perturbation prediction, where, like I said before, with the sort of the train test splitting regime that or method that we're using, for a given data set, you actually have multiple train test splits of increasing difficulty. So you get a data point, you get a sort of performance metric. In our case, we quantified it using the mean squared error. And you can see it here nicely in this sort of visualization. At every sort of sparsification probability, you get a certain mean squared error. And then as your train test split gets more difficult, your mean squared error degrades. And then the rate at which sort of in our head, sort of the best way to sort of anchor and interpret our models is to sort of look at how quickly your baseline degrades compared to how quickly your model degrades, and then sort of calculating the area in between these two graphs to sort of give you a holistic indication of how robust your model is to distribution shift, which is a very often cited problem in this field and many adjacent fields. So actually, this is sort of two metrics for one. You calculate your MSE for each and every one of these points, and then you can get your AUSPC out that indicates to some extent the robustness of your method, if there is any at all. Furthermore, we use the e-distance to quantify perturbation strength. So this is a sort of based on the e-test to sort of make this statistically significant, running many tests to check whether your perturbed samples of cells and how far away these perturbed cells are from your control, to sort of quantify exactly how strong your perturbation is, how far has your perturbation pushed the cells out of their control state, right? And then the e-distance allows you to quantify this. Lastly, we use the cosine similarity between pre-training data sets and the fine-tuning data sets. So of course, with each of these single cell foundation models, they have been trained on some sort of pre-training data set. And there has been previous evidence in the context of cell depth annotation provided by Cui, who developed SCGPT with his collaborators, that actually the similarity between pre-training and fine-tuning data very much are indicative of how well your fine-tuning stage sort of improves upon for your base pre-trained task. yeah, so we wanted to add that in as well to see if we could find similar findings in the context of perturbation prediction. So for experimental setup, there are sort of two that we look at in this work. We firstly do the sort of standard one in which you predict across the entire 2000 highly variable genes transcriptome. So a pre-processing step with single cell data is of course selecting highly variable genes. The idea being that, in these single cell RNA sequencing experiments, you measure thousands and thousands of genes, their expression value for any given cell. Not all of them are all that interesting or variable. And so you just filter those out to, generally speaking, you take the top 1,000 or the top 2,000 highly variable genes. We will see that actually for perturbation prediction, this is already sort of a gross overestimation of what is interesting to look at, or what is really very measurable to look at. Maybe let's put it like that. And so next to this, we also look at the top 20 differentially expressed genes for given perturbations, which are arguably sort of the most interesting ones for a given perturbation. And we will see this back in our results as well, actually. So talking about the results, let's have a look at them. First of all, these are our results for the predictions across the 2000 highly variable genes. And just at a glance, specifically looking here at the AU-SPC bar plots, I think it's very obvious that, predicting across the 2000 highly variable genes, there's sort of a lot of homogenization going on there, sort of suggesting that, maybe many of these genes are not all that signal rich for this particular task. That being said, with our sort of evaluation setup of the sparsification probabilities and spectra, and having these trained test bits of increasing difficulty, there is this notable trend of performance degradation that is particularly visible in the foundation models and the mean baseline when it comes to this highly variable gene, when you look at it from this highly variable gene angle, whereas the MLP baseline seems to suffer a bit less. And you can see that this also reflects back in the AUSPC metric, where the MLP baseline performs best, although, yeah, the variability here is not too cost significant. And to look a bit closer at this, like I said before, you want to look at actually the top 20 differentially expressed genes, which are sort of the most interesting ones for a given perturbation. So you can do this for all your perturbations. You look at the top 20 differentially expressed genes, and now you sort of restrict your evaluation to only these. And this is where you start to see a bit more variability in your results. Interestingly, again, the mean baseline is already quite performant, still suggesting, that even though there might be a bit more sort of interesting signal going on this scale, there still is a lot of sort of averaged out signal present, and we will see this later on a bit as well. However, foundation models just sort of restricting to the positive signal, right? Restricting to the actual genes that change seem to perform, or at least a subset of them, seem to perform slightly better than this mean control. Here's the delta AUSPC, where you take the difference of your performance with the mean, where a positive value suggests better than mean, negative suggests worse than mean. And while all in all, it's not that impressive, especially considering the amount of compute that goes into a foundation model compared to the amount of compute that would go into a mean baseline, I think we can all agree that this is not necessarily, this would not necessarily argue for using a model specifically for this task. So then this begs the question, given these results that are not all that impressive, they're not all that favorable for foundation models, to be completely honest. What's going on here, right? We want to look into this in a bit more detail. And so we can go back to this metric I previously introduced, the e-distance. And it's interesting to look, sort of we hypothesized that, a lot of this data we have, which are single and two gene perturbations, it's all well and good, but generally speaking, just perturbing one single gene, that this might not be essential, might not be very central in big gene regulatory networks, that might not elicit, very strong perturbation effects, and in turn, sort of strong signal that your model can learn from. And indeed, this is true, because kind of most of the perturbations here cluster in the low e-distance regime. And this also suggests that, at training time, your model mostly sees sort of very modest average, if you will, perturbations. As would argue for, a more even representation of perturbation space would be beneficial to also be able to, predict the bit more out there perturbations that oftentimes are the ones that are actually interesting. There are some outliers, some interesting cases, and oftentimes looking at these cases gives you some more ideas about potential failure modes, potentially what might be going on. So we'll be doing that shortly. Another thing to take into account with the spectra method is sort of the sample size of your data. As you might imagine, as you increase the sparsification probability, increase the number of subgraphs in your similarity graph, at some point you cannot really sample anymore from these subgraphs because they're just too dissimilar. Normally you would have this sparsification range between 0.0 and 1.0. In our case, after 0.7, sample sizes already became too few to meaningfully predict with them. So this is sort of a caveat to bear in mind when using a method like spectra. Okay. Like I said, let's have a look at some actual specific examples to see if we can, make sense of it, and get some hints or some clues as to what might we be missing to successfully predict on these tasks, and is it even reasonable to assume that then foundation models could get us there? So here we have CEBPA, which, if you look at the MSE, is actually quite reasonable compared to the very perturbations with a relatively low perturbation effect, but it's predicted quite accurately, while it's like by measure of the e-distance sort of the most strong perturbation. And now where does this come from? In this case, we can see that particularly the top two perturbations are sort of very strongly affected, and then very quickly the effect drops off to average, like zero change between perturbed state and between the control state. So okay, this is sort of one perturbation type that you might have, in which you have very concentrated effector at the top that gradually sort of tails off to average. And now you can imagine that all the average genes here, they are predicted relatively well, as suggested by the mean control and the fact that even the MLP and using the foundation model embeddings seem to mostly learn sort of the average effect on aggregate. So that kind of explains that observation. Whereas on the other end of the spectrum, you might have a gene like CEBPE, which also has a relatively high e-distance and is poorly predicted, especially compared to the other predictions. Now, here we can see a completely different distribution. Like this one is all over the place, especially compared to the next one. You have tremendous impact here on like however many, like 10 genes that get upregulated significantly. And then here we have sorted the differentially expressed genes by absolute magnitude. And so we jump over to like 4 genes that actually get downregulated relatively significantly. And the model is guessing around the average, because that's of course what it's seeing most, and completely missing the mark there. And here similarly, another sort of type of distribution you see where it sort of evenly drops off, and you have some average genes in there as well. But just in this ACI-ZF3 case, again, very interestingly, the models do predict some, with high variability, some sort of significant effect. So suggesting that they have sort of, based on this distribution, been able to pick up, okay, with this distribution, there are genes that are significantly affected, but it is just clueless as to what the direction is and the magnitude of this change. And this also manifests in the sort of the big variability, even between different models. And so I think these are sort of three different failure modes that all kind of point in the same direction of perturbation space being just too, heterogeneously represented in favor of very modest average perturbations, and embedding then your data with foundation models, even though on a sort of cellular level that might give you some interesting substrates to work with, looking at perturbation level that does not really push the needle any which way. So that is kind of the current state, what we're dealing with. So yeah, let's just sort of summarize what we've seen so far, the key takeaways. So most importantly, I think for the project that we presented here, PertEval-scFM, we have sort of proposed sort of systematic toolkit that you can apply to benchmarking whatever model. Like we applied it to foundation models. It equally applies to more bespoke specific models for perturbation effect prediction, consisting of evaluation metrics, a train test split, simulation metric for distribution shift, simulation, and sort of assessing the robustness of your predictions, as well as some sort of interpretation methods to look at how the model is doing, where it's going wrong, and also sort of contextualizing this with the e-distance, sort of different metrics and suggestions that altogether we think form a solid foundation to really look at these perturbation predictors in the scrutiny that it needs. Sometimes you see people just using a simple correlation metric to say or to make claims about whether perturbation prediction method is good or not. This is a very generally a very tainted view, actually sort of misguided view of actually what's going on. And so what we found in the end by applying this method to zero-shot foundation model embeddings is that they do not anywhere near systematically outperform baselines, especially considering compute. It's arguably not recommended to use them in this case. And the metrics we've applied highlight specific challenges that these zero-shot embeddings do not provide any more robustness to distribution shift as you might already expect from baselines. And by looking at your predictions together with the e-distance, suggests that the model struggles very severely with strong and atypically distributed perturbation, sort of, begging the question of improving on the data side of this modeling problem. So currently, what's next for PertEval-scFM? Currently, we are working together with great people over at the Marinka Zitnik lab, led by Alejandro Velez, who oversees the Therapeutics Data Commons, the TDC, in order to integrate our framework into their very holistic framework. With like they have tons of different interesting prediction tasks and standardized data sets and evaluation metrics, which we're very happy to soon be a part of. And also future directions sort of based on what we've seen here in this benchmark, as well as the many other slew of benchmarks that are similar to ours that came out, suggest there are some significant improvements to be made for these models if they are to be successfully applied to perturbation prediction. In the case of, for example, representing perturbation in silico, like we have seen how we did that in our case with sort of the nullification, there's a lot to be said there. And already taking this into consideration at sort of the pre-training stage, with more sort of bespoke perturbation foundation models would arguably help a lot compared to sort of the current state of affairs. And this goes hand in hand with sort of developing a more sort of bespoke training scheme specific for perturbational data. Yeah, and then in line with that, so what our work suggested, like I said before, is that the perturbational space is not equally represented across perturbations whatsoever, mostly favoring very average and modest perturbations. And so what we want actually is, on the one side, having data that spans the full range of the human phenotype, if you sort of see, oftentimes we think of cells as these very resolved entities, like it's cell type A or cell type B, but in actuality, in actual living systems, this is sort of more a continuous spectrum, and you just take a snapshot of this continuous spectrum, and the cell might be changing its identity, or it might be going through some other sort of process, which you cannot fully appreciate with just such a snapshot. In addition to including like multiple modalities of biology and not just RNA or the transcriptome, together with clinically relevant cell types. So yeah, we argue that on the data side of this modeling problem, there's a lot of ground to be covered to make them better. Interestingly, this is actually a contested point. There are people that say we have plenty of data, we just need to scale compute. Yeah, so this is by no means a decided and set thing, but this, given what I've seen, I think is sort of a logical way forward. And then, of course, sort of create more bespoke models. We did some testing with gears, which is sort of a graph method that introduces some inductive biases on how you represent perturbations. And it already showed in our eval a lot better than any of the baselines and any of the foundation models. So that's also sort of an interesting find for improvements. Finally, I'd like to thank all the collaborators. This was a tremendous team effort with many collaborators across many different institutes. I'd like to thank my supervisors, funding bodies, and in particular the QMOL ITS Research and Software Engineering team that are always very, very helpful with running and debugging experiments on our cluster. And yeah, thank you guys for your attention. And if there are any questions or discussions, I would happily entertain those now.
Thank you, Aaron, for the wonderful presentation. We now have time for questions. If you guys have any questions, please feel free to raise your hand or write it down in the chat. It looks like we had a question in the chat, but it may or may not have been answered during the presentation. Why are we using the Pearson correlation for the perturbation matrices?
Right. Okay. I heard a message come in.
Yeah.
Okay, let me just try and answer this one. So the Pearson correlation, the motivation behind it was where we wanted to have some sort of first order approximation of genetic interactions between the perturbable gene and all the other genes present in the cell, so that if you want to generalize to unseen perturbations, your model has already some sort of notion of interactions between genes. Whereas with a one-hot encoding, you literally only tell your model the identity of, so this is the gene we're perturbing, and you're not telling it anything of how that might interact with nearby other genes. So that's the sort of the rhetoric why you want to use a correlation here over a simple one-hot encoding.
So this correlation is per gene, or like,
yeah.
For example, if you perturb one gene, then you would have one Pearson correlation. So you just replace the raw expression with a Pearson correlation, or you add it?
No, you modify it. No padding going on. It's per gene. Yeah.
Okay, thanks.
No worries.
Wonderful. Liam?
Yeah, thanks. Could you go to slide, I think it was maybe slide 15. I'll see if it's the right one. Yeah, perfect. So yeah, I just wanted to check I was understanding this right. So this is showing, like the left plot is showing for the top differentially expressed genes, the true differential expression in the black line, or the mean differential expression, and then the dots are the predictions.
Yes.
So I guess first question is, were you surprised to see how consistent the how consistently incorrect the predictions were across the models. Like it seems like they all kind of predict the same wrong value
Yeah,
or off value.
I can't say I'm particularly surprised by it, especially this is of course sort of retrospectively, right? But especially in retrospect, looking sort of at the data that's in there, I don't think I was very surprised. Like the big question was that we wanted to answer at the outset is, does this pre-training and generating these cell embeddings sort of inject any sort of additional helpful semanticity into your embeddings that would allow you to do better perturbation prediction? And I think the fact that all of them collapse to the mean, which is kind of what you see here, answers that question to no. So in retrospect, the answer is no. Maybe not so obvious when we started it out, but yeah.
And so then, does this, of those top differential express genes, does that include the one that is the target of the CRISPR guide, like the one that's knocked down or knocked out?
No.
Or is that one omitted?
That one is omitted. So these are only the ones that sort of downstream affected by perturbing that one particular gene.
Okay, gotcha. Okay, yeah, because I was thinking that the top differentially expressed gene should be I guess a large down regulation if it's knocked down or knocked out. But if you're omitting it, then that makes sense.
Yeah.
Okay, thanks.
All right. Thank you for your great question.
We have space for a few more.
Yeah, I have a question here from the room. Hi, Aaron. You can hear me?
Yes, loud and clear.
Good. You had a slide towards the beginning about the three sort of assumptions that were made in using like these foundation models. Yeah, that's right. That slide here. So I was wondering about the one to the right, the MLM objective, right? Being the one that's used to model gene expression, for instance, right?
Right.
Now, what you're doing that study is that you're saying, well, if we're perturbing the latent space, essentially, or shutting down whole genes, it should then appear in the ability of the model to reconstruct the gene expression for that gene, right? So that reconstruction should be perturbed. And from my understanding. And then at the end of the presentation, you presented that, well, perhaps we would need more specific pre-training strategies for perturbation data.
Right.
Right. So did you have in mind what kind of, well, you probably thought of this already, like what kind of objectives that would be? And would it still involve some form of gene expression reconstruction, or would it be like a more stark kind of divergence from that paradigm?
Yeah, that's a great question. And we did do some high level thinking on this, like not really into very much detail just yet, but I can share the thoughts that we had there. I reiterated at some point as well that it was very important how you represent perturbation in silico. There are different types of perturbations. You can activate genes, you can upregulate them, you can downregulate them, and this all very drastically differently changes what would happen to your transcriptomic space. And currently, in the current sort of iteration of these foundation models, there's no sort of notion of directionality or type of perturbation. So this is, I think, the lowest hanging fruit if you want to improve this. So you could keep kind of the pre-training the same, but you just have kind of different tokens for different types of perturbations, for example, which would be sort of a very first low hanging fruit step. Yeah, beyond that, I haven't thought of any sort of very structural objective changes as to away from master language modeling. But arguably there might be some stuff there as well that would be interesting to look at.
Yeah, like, and something else you point out that's interesting from that third figure is the fact that, yeah, when we're doing MLM here, it's gene interactions across cells, right?
Right.
When you're perturbing, you could think as perturbing the cell itself, and then it's the gene expression profile that changes, right?
Right.
So you couldn't really have one cell unperturbed really tended to another perturbed cell and have that make any sense. So would we also have to change how the different types of input features that are actually trafficked into the model to start with?
Yeah, definitely. And I don't think that's a trivial thing. Like, you could present it as such, where indeed the multi-language modeling objective learns also from, for example, similar cell types across cells, which to my understanding is, as a lot of them do it, but it's not necessarily the only way in which you could do it. You could restrict that, or even lessen those constraints a bit, make them a bit weaker to include maybe more different cell types. And if you so currently these foundation models are pre-trained on data sets, again, mostly using sort of very rigid cell type annotations. And whereas with perturbations, you kind of move away from this paradigm of cell types, and you get into this intermediate stage with, I really like the Flag science paper that goes into phenoscapes. I would really recommend anyone interested in this reading this, what is a cell type paper. He also goes into this actually, and I really like that, sort of more that way of thinking about it. And then when we think about how to sort of get a sort of pre-training objective, having all these sort of biological notions that are very important that are presented here in there as well. And so you kind of move away from cell types more towards sort of cell states, maybe, or phenoscapes, as they call it, in this particular paper.
Well, yeah, actually, when you showed this, I was all in on this kind of con, because I think other people have questions. But when you showed this article, I read this a while ago, and I think you mentioned reprogramming transcription factors. So arguably, you could think of like in some occasions as perturbation that's actually being transitions between different cell types, right?
Yeah.
So if you have one cell type that's more differentiated, right? Well, you could maybe knock it back into its more precursor forms.
Yeah.
So the perturbation could actually be in line with changes in so-called cell types, right?
Yeah.
Because
Yeah.
cell types aren't just these fully distinct entities, as you're kind of demonstrating with referencing this article. So
yeah, definitely. And also, like, I mostly work in drug discovery, so for us it's always often interesting to think about disease states and healthy states, but like the reasoning there is again similar. Yeah, 100% agree.
all right, thanks. Awesome presentation.
Thanks.
Professor Lemieux?
Yeah, hello, Aaron. Thanks for the presentation. It was really interesting. I'm wondering if when you compare those different models, are you in a situation where the noise of the output you're trying to predict so high that the difference that you see is just, capping a sort of upper bound in the quality of the prediction? Do you have any thoughts on how to test for that?
Ooh, that's a great question. Thoughts how to test for that?
Yeah, maybe I can just ask another question just before.
Okay.
So you're predicting gene expression in a single cell, or are you pulling together the profile of multiple cells that receive the same guide?
Yeah, the last one. So we sort of cluster them by perturbation. So, and that's already in and of itself sort of a noise reducing strategy, right? But not fully. There's still, this prediction problem is plagued. And ironically, I couldn't go into all great technical details, but for that, please do consult the paper. Another thing we do is sort of on the input side is sort of pseudo bulking, where you have single cell data, but in the intercell variability is just so high that just challenges your prediction task so much so that it's been found empirically that the best thing to do is to pseudo bulk your single cell data. So
so I read.
Go for it. Sorry.
I read in the paper that you pseudo bulk the baseline, the no perturbation baseline, but you do it also on the perturbation side.
Yeah.
Okay, cool. So I'm less concerned about the noise in that context.
Yeah, but it's a great point. It's a great point to bring up. And also with these foundation models, actually, there have been claims made that they are sort of regularizing against this type of noise. Again, reinforcing the thought that maybe, those embeddings they generate would be a bit more robust against that, and in turn perform better substrates or be better substrates to do predictions with. not the case. It turns out, or at least our work suggests that that's not as currently constructed yet the case. But yeah, very important and good point. So thank you for bringing that up.
Cool. Thanks.
Any more questions? If there are no more questions, let's take another, like a few more seconds to thank our speaker again. And that's the end of the recording. Thank you very much. Oh, never mind. We have a question. Yeah, please.
No, sorry, it was a mistake. Thank you for the presentation.
Wonderful. So then that stops the recording.
That's a good
Everybody,
question.
yeah. Have a nice day. Thank you.
Yes, guys.
Thank you.
Thank you.